just saying that, as Paulo said, I'm a newcomer here at CIBIO-BIOPOLIS. I was a CNRS, a senior CNRS scientist at the University of Lyon until last August. And then I joined the center here. Okay, so my presentation will be about sexes and sex chromosomes in plants. Why is it important to study separate sexes in biology? This is because the existence of males and females, the differences between males and females, how sexes are determined, and the consequences for the species are major questions that are really at the heart of biology, and that are connected to many fields in biology. Of course, males and females, they are very common in animals, and we tend to forget that they also exist in other important groups, and they also exist in plants. There are about 15,000 species of angiosperms that have separate sexes, males and females, and they are called dioecious plants. You don't hear a lot about them because it's considered a minor sexual system in plants, simply because the major one is hermaphroditism. Most of the angiosperms are hermaphroditic, as you know. They have bisexual flowers. But nevertheless, 15,000 species, 15,000 of dioecious species, it's a lot of species. If you think about it, it's about the same as all the mammals and birds combined. In angiosperms, dioecy is of recent origin. The ancestor of all angiosperms was hermaphroditic with bisexual flowers. And phylogenetic studies have shown that there were about 800 to 5,000 independent evolution towards dioecy. In animals, we have information about how sex is determined, sex determination, for more than 90% of the species. Now, if you look at dioecious plants, we have rough information that is whether sex determination is genetic or environmental for about 3% of the 15,000 species that I mentioned earlier. So very different situation. In animals, the most common way of determining sex is through sex chromosomes. It's the case in our species, but also in numerous animal species. In plants, most of the species for which we have information on sex determination, they have sex chromosomes. But we know very little on these sex chromosomes, and also we don't know, because it's so few species, we don't know whether sex chromosomes are widespread or not among dioecious plants. So really there is a very different situation when it comes to sex determination in animals, in plants. In animals, we know a lot of things. In plants, we know almost nothing. So in this phylogenetic tree of angiosperms. There are orders where you find species with sex chromosomes. I guess you cannot see anything because it's too small, but there are many orders that in which you can find sex chromosomes: cucurbitales, rosales, brassicales, vitaceae, caryophyllales, and others. They represent about 40 different systems out of possibly, as I said, 800 to 5,000 ones. So again, we have very little information. This is a sketch to show you the scenario that we have for the evolution of sex chromosome in plants. We think that the sex chromosomes, they originate from a pair of autosomes on which the sex-determining genes emerge, and then recombination is suppressed in this region, in the sex-determining region. The non-recombining region tends to grow during evolution. So this is the regions in green and red in the picture, and they can grow very large, and then in that case the sex chromosomes become heteromorphic. They become different, and the X chromosome is not recombining in males, but it's still recombining in females. The Y chromosome is not recombining at all, and it degenerates because of that. And first, it grows very large because it accumulates transposable elements, and then it tends to lose genes and to become very degraded, and it becomes shorter than the X chromosome, and it can even disappear. So this is the scenario that we have for plants. It relies on very little case studies, and it is mainly inspired from information from animals. So this scenario is extremely fragile. Why is it important to study plant sex chromosomes? Because if we want to understand the mechanisms of sex determination and how they evolve, we need to have a better knowledge of those mechanisms in this important eukaryotic group that plants are. Second thing, dioecious species, they are overrepresented in crops. About 20% of the crops, they are dioecious or derived from a wild ancestor, a wild progenitor that is dioecious. I put on this slide a couple of examples: papaya, kiwi fruit, grape, date, and so on. They are dioecious or come from a dioecious progenitor. Of course, if you don't know, you don't understand sex determination in those plants, it's a problem if you want to control reproduction. For example, in the many dioecious crops, only one sex is economically important. So you need to, the knowledge of sex chromosomes, they allow identifying plants with that particular sex really early and to increase the output. Second thing is when you know sex determination mechanism, you can manipulate it, and this can be really useful to obtain new varieties because you can control crosses. The problem is the Y chromosome is a bioinformatic nightmare. So it's very difficult to sequence a Y chromosome, and more importantly, to assemble a Y chromosome for two reasons. The first reason has to do with the standard approach that we have in genomics, that is whole genome shotgun. Maybe you have heard about it. The idea is to fragment a genome into pieces and then use sequencing technologies that will produce small sequences called reads. And the next generation technologies, they give even smaller reads. And those reads, they are like pieces of a puzzle. Then you have to put it in the right order to reconstruct the sequence of the chromosomes, right? And this step is called assembly, and it's a very well-known problem. When there are repeats in a genome, this is going to make difficult the assembly, because the repeats, the different copies of an element, they tend to collapse to assemble together. And as shown here in this picture, and this leaves gaps in the assembly. Okay, so you get a fragmented assembly. You get, instead of having one sequence for a chromosome, you get several sequences. The Y chromosome is full of repeats. So with this approach, it's virtually impossible to assemble a Y chromosome. The second problem has to do with the coverage. So if you sequence a male individual, a XY male individual, the coverage for the X and the Y chromosome is going to be half that of the autosomes, simply because there is one copy of the X and one copy of the Y. And the quality of the assembly has to do with the coverage. So if you sequence a XY male individual, the assembly is going to be worse for the sex chromosomes compared to those of the autosomes. And this actually happens for the Human Genome Project. They sequence males, and the assembly of the X and the Y were terrible. So they had to redo them again. So after that bad experience, all the Genome Project, they tend to sequence XX females. So if we so in the field of sex chromosomes, we had these technological problems, and so we had to come up with alternative approaches to study these chromosomes. Different groups have developed different methods, and on these slides there is a list of methods. I will come back to that list later, but today I will explain the method that we have been developing in my group in Lyon to study sex chromosomes in non-model organisms, and also how we applied those methods to dioecious plants. Okay, so there are several tools, and they are part of a suite that we call SEX-DETector. So this is the first tool, SEX-DETector, and I'm gonna explain the principle. You will see it's very simple. So the idea is to study how alleles are transmitted from the parents to the progeny. So for example, here the father is big A, the mother is small a. All the offspring, they are big A, small a, whatever their sex. So clearly, this marker is autosomal. Okay. Now, if you have the father big A, small a, the mother small a, and all the sons are big A, small a, and the daughter small a. So the big A allele is transmitted from father to sons. This is XY gene, XY marker, right? We can also identify X hemizygous marker, in which the father is big A, mother small a, and all the sons are small a, all the daughters big A, small a. So this is the hallmark of X linkage. Okay. So these markers, they don't have Y alleles. So this actually the first to use this approach were these people, Dmitry Filatov and Deborah Charles in a 2011 paper, and they used an empirical method to perform this analysis. So what they did is they obtained RNA-seq data for the parents and a couple of plants from the progeny. And they used this RNA-seq data to genotype the individuals. I'm going to show you how we can do it later. And for all the markers, they looked whether the patterns of transmission was autosomal, XY, or X hemizygous. The problem is genotyping data are data, and there are errors in data, and their way of dealing with that was not satisfactory. They used arbitrary cutoffs. And so in the group, we decided to develop another method. So this was the PhD of a very bright student in the group, Aline Meul. And the idea was to develop a method based on a model that would describe the experiment. And this model had parameters that were estimated using the data through a maximum likelihood approach using stochastic expectation maximization algorithm. And then the method would return posterior probabilities of being autosomal, XY, or X hemizygous for the different markers, SNPs. And these probabilities were then combined to get a global probability for a gene. So as I told you before, the experiment is really easy. You need to have a cross, parents, progeny. You sequence a few individuals, both parents and a couple of less than 10 individuals in the progeny. You get RNA-seq data. You can, if you don't have a reference genome, you can assemble a de novo transcriptome using the RNA-seq reads. Then you can map back the reads of each individual onto the reference, and you do the SNP calling. And this way you have the genotype of all individuals for all SNP in your transcriptome. And then the genotyping data, you can run SEX-DETector on them and get inferences on sex linkage. Sometimes we realize that it's not adequate to use RNA-seq data for some species, for some systems. In particular, when the non-recombining region is really small. So in that case, it's better to get genome data, and we developed a much faster version of SEX-DETector in C++ to do that. So this was done by François Gendreau, who is a computer scientist and a C++ master. And so we got a C++ version that is 600 times faster than the original version. The last version that we developed is called SEX-DETector in population, because for some organisms, it's not possible to obtain a cross. So the only way to study sex chromosomes is to go out on the field to collect biological material from male and female individuals that you can find in the field. And this method is really meant to analyze such data. The framework is the same as for the SEX-DETector version, older versions, but here the model is completely different. It's a population genetics model. So we can have we can actually compute the expectations in terms of genotype frequencies for given Hardy-Weinberg equilibrium for autosomal XY and X homozygous markers, and we can compare the theoretical expectation with the observations, and using the same probabilistic approach, we can then detect what are the sex-linked genes. Okay, so these methods, we have applied them to a number of dioecious plants. And today I'm going to focus on 3 examples: Silene latifolia, a wild plant, and Cannabis sativa and Vitis sylvestris, 2 crops. Silene latifolia is a Carliophyllaceae. It's called white campion, and it's a very common plant. And it's a dioecious plant with XY system. It's a very well studied dioecious plant since Darwin, actually. And it is the system is quite young, 11 million years old, but it's already heteromorphic. As you can see here, the Y chromosome is larger than the X chromosome. The problem, it's a very nice system. The problem with this plant is the size of the genome. It's a very large genome. It's 3 gigabases long. It's repeat rich, and the X chromosome is 400 megabases long, and the Y chromosome is 550 megabases long. It's a huge Y chromosome. It's the size of, for example, the grapevine genome is the same size. So I think it's the largest Y chromosome documented so far. What we did is we use SEX-DETector. So we sequenced parent and progeny from across. Because we don't, of course, we don't have a reference genome. We assembled a de novo transcriptome. We did all the workflow that I mentioned earlier, and we obtained these inferences. So we had 13,000 autosomal genes, 1,000 XY genes, and about 300 X homozygous gene. We tested whether the quality of these inferences using a tester set of 129 known autosomal and 80 known sex-linked genes, and we obtain a specificity and a sensitivity that are very high. In particular, the sensitivity, 83%, is much larger than the sensitivity of the empirical methods. Okay, so then we studied the sex-linked genes in this species, and we found that actually, if we compare the expression of the X allele and the Y allele of a given sex-linked gene, we found that actually the expression of the Y allele is reduced compared to that of the X allele. So this here on this graph, you have the Y over X expression ratio, and you can see that a lot of sex-linked genes have a ratio that is lower than 1. And the mean is actually significantly lower than 1. So this means that in principle, the males, they should have a problem of underdosage for all those genes, right? And in animals, we know that this problem is solved by a mechanism called dosage compensation. Maybe you have heard about it. Maybe not. So but in plants, it was not known whether dosage compensation existed or not. So here I'm showing the analysis that we performed. We looked at the expression of X in females, expression of X in males, expression of Y in males, combined expression of X and Y allele in male. So in red on this graph, you have expression in female, and the other colors represent expression in male, and for different gene categories. So the first category has weak degeneration. The last category has very strong degeneration, and the level of the degeneration is in between for the other gene categories. And you can see that actually the expression of the X in males, without dosage compensation, it should be 0.5, okay, for all the categories, simply because there is two copies, there is only one copy of the X in males, okay, and two copies of the X in females. So the expression of the X in males should be half that of the expression of the X in females, okay? This is not what we observed. What we observed is that the expression of the X in males increases, and as the level of degeneration increases, as the level of Y expression decreases, right? It's very clear on this graph. And so now if you look at the expression, the combined expression for all alleles, you can see that the expression is actually the same in males and in females for the different categories. So this is evidence for dosage compensation. There's one category that was strange. It's the last one. In the last one, the signal was less clear. The last one represents the X semizygous genes. And actually, we know that this is one of the drawbacks of our method. It's easier to identify the XY genes compared to the X semizygous genes. They are more difficult to identify. Later, another group actually studied these X semizygous genes using a draft genome, a partial genome of Silene latifolia, and they had an unbiased set of X hemizygous genes. And when they looked at their expression, they found that actually those genes, they have two populations. There is one population of genes without dosage compensation, and there is another population with full dosage compensation, looking at the expression of X in male compared to expression of X in female. Okay. And in another study, Aline looked at the expression in an outgroup without sex chromosomes, and she performed an allele-specific expression analysis, and she found that actually, if you compare to the outgroup, the expression of the X in male has increased. Okay, it is the red bar is larger than the purple bar in the outgroup. So compared to the ancestral expression has increased in male. The big surprise was in females. If you look now in females, you see that the expression of one of the X is actually also increased, and it's always the X from the mother. So it means that actually the mechanism for dosage compensation in this plant is based on genomic imprinting. It's the X from the mother that is modified and overexpressed, and this is true for males and also for females. So actually in this species, the females have a problem of overdosage. And this is very interesting. Maybe it has to do with the fact that the system is young. So this is to recapitulate our findings. There are different dosage compensation mechanisms in plants, in life, in animals. So in flies, for example, it's a straight overexpression of the X only specific to males. In mammals, it's an overexpression of the X in both sexes, and then inactivation of one of the X in female through X chromosome inactivation. I'm sure you heard about it. And in C. elegans, it's overexpression of the X in both sexes and downregulation of the X of both X's in hermaphrodites. C. elegans has hermaphrodites and males. In Silene latifolia, what has been so we found something that resembles actually what happens in marsupials. So in marsupials, the X chromosome is overexpressed in both sexes, and then the X from the father is downregulated, is silenced. Actually, in this plant, the X is not silenced, but another group actually found that there are positive marks on the X in males and one of the X in females, and negative epigenetic marks on the other X in females. We found more species with partial dosage compensation, more plant species that are listed here. So it seems that it's not something weird in Silene latifolia. It seems that dosage compensation does exist in plants, and it is possibly widespread. And also the big surprise was to find it in such a recent system, because from the studies in animals, people thought that it was really difficult to evolve dosage compensation mechanisms. And because it was found only in all systems, and we found actually a dosage compensation mechanism in 11 million years old system. Okay, so I think I have only 10 minutes to tell you about the other stories. So I think we'll have to make a choice now, whether I speak about grapevine or cannabis sativa. I think I'm gonna go for grapevine because I will explain why. Okay, so the grapevine story is also interesting. So in grapevine, we know that the wild progenitor of Vitis vinifera, that is called Vitis sylvestris, is dioecious. It has a XY system, but something has changed in grapevine during domestication because cultivated grapevine are hermaphroditic. Okay, we know that the sex locus, the locus containing the sex-determining genes, is on chromosome 2. There are 3 different haplotypes, X, Y, that are found in the wild grapevine, and YH, that is a modified version of the Y haplotype that is found only in domesticated grapevine. Even some candidates have been proposed, but before we started our study, the sex locus boundaries, gene content were not precisely known, and there was no model for sex determination in grapevine. So what we did, first we sequenced a genome of the wild grapevine, Vitis sylvestris, and then we tried to compare our data with Vitis vinifera. So as I told you, we first obtained a female Vitis sylvestris genome. We used PacBio technology, more than 100x coverage, and so we got a very high quality genome with a chromosome level assembly. The grapevine genome is not that difficult. It's only 500 megabases, but still, that was not that easy at the time. And then we obtained a cross of Vitis sylvestris. We sequenced the parents, 5 sons, 5 daughters, using resec approach, so genome data, and we run SEX-DETector ++ on the data. And we actually got very nice results. So this is chromosome 2. And so these are different ways of showing the sex-linked genes. We found a very clear peak here for the sex-linked genes. Then we obtain back sequences for the X haplotype, the Y haplotype, and the YH haplotype. And we could, and then we could look at gene content of the different haplotypes and expression patterns. And we found something really interesting. So this is the X haplotype. And we found that one gene called IMP1 is actually present on the Y and YH haplotype, but it's not on the X1. And this gene, it's deleted, partially deleted on the X1. And this gene is very interesting because it's a pollen gene. It's a male gene. We also found 3 equally interesting genes that are shown in blue. because they have very interesting expression pattern, especially this one, APRT3. So this is the expression in cultivated grapevine, in male and female wild grapevine during floral development. And you can see that only in males, this gene is overexpressed, highly expressed at the end of the development. And it's not in hermaphrodites, not in females. And it's very interesting because this gene is a cytokinin repressor gene, and cytokinin is a phytohormone that is known to actually stimulate the development of the female organs. So it looks like a very good candidate to explain why the males, they don't develop female organs. Okay, so then we came up with a model for sex determination in grapevine. So we think that in the ancestor of the Vitis genus, the sex locus was actually autosomal, and then some sex-determining mutations came up, recombination was suppressed, and so on one side, on the X haplotype, IMP1 was deleted. These three genes, they kept the original expression pattern. On the Y haplotype, IMP1 remained the same, remained functional, and these three genes, they evolved a new expression pattern. Also, because the Y haplotype is not recombining, it accumulated transposable elements, and it's very clear. We found a lot of transposable elements in the Y haplotype. It's also why we struggled so much to actually assemble the Y haplotype. And before domestication, two mutations of the Y appeared. A first mutation was modification of the expression pattern of APRT3. And we think this was because there was an accumulation of transposable element in the cis regulatory region of that gene. And the other mutation was actually a recombination between the X haplotype and the Y haplotype at the APRT3 region. Okay, so this is what we found when we looked at several YH haplotypes. And the interesting things, so this is to tell me that it's over. The interesting thing was that when we compare the Y haplotype and the YH haplotypes, looking at the substitution patterns, we found that actually we could estimate the age of the YH haplotypes, and a surprise came up because we found something that is much earlier than domestication. So what we think is that actually these mutations were standing variations. They were already in natural populations, and the first grapevine breeders, they actually picked those mutants in the population because they were convenience, okay, to get fruits. Okay, so this story is also very interesting after drink and now it's drug. But I'm not gonna tell you about it because I'm running out of time. But please come back October 29th, because Jivan Prantou, who is a PhD student working on that project, is going to be here in CIBIO and is going to present his work, okay? So I'm gonna go really fast here and just go to jump to the conclusions, because I want to give you some take-home messages. The first one is, if you are interested in sex chromosomes, you should consider these tools that I mentioned, because the experiment is really cheap and the tools, they work fine. And so you can have very important information on sex-linked genes in any organisms. The approach is really good for plants, simply because actually these methods, the segregation analysis methods, they work better for young systems. So when the divergence between sex chromosomes is not too high, okay? If you work on a system that is old and very diverged, you should go for these other methods. And if you don't know, for many organisms we don't know, you should combine those methods. Okay, so these methods, they have been really useful in plants. So I told you about the work that we have done, and actually only part of the work that we have done, because so this is all the species that we have studied are listed here and here. Sometimes we are different group studying the same species. This is the case in grapevine. And I should mention that the model that I presented to you, not all the groups agree with it. Okay. No, it's important. It's I forgot to tell it before. And the other groups, they have been working on those plants, and you can see that that these methods, they have really allowed great advances about plant sex chromosomes. This is the list of all the sex-determining genes that have been found in plants. It's amazing if you think about it. The first sex-determining gene was found in 2014. Okay, so now we have the sex-determining genes for about 10 systems in about 5 years. This is amazing. And it's so fast that actually I cannot tell you more about it, because there was no synthesis with all those data. We are still figuring out how to interpret the data that we found in the different systems. The last thing I wanted to tell you is that those methods, they are going to be really useful to look at other groups where we separate sexes and sex chromosomes. So I told you that in animals, we know a lot of things. In angiosperms, we know very little, but if you look at gymnosperms, bryophytes, it's worse. If you look at green algae, red algae, brown algae, it's even worse. We know almost nothing. And these groups, they are eukaryotic supergroups. They contain a lot of species, and there is a lot of work to do. And okay, so now I just wanted to thank all the research that I presented to you comes from this group in Lyon. It's a group that I created a couple of years ago, and I wanted to thank all the members of the groups. I am no longer part of this group. One thing I'm really proud about this group is that 4 of the former students and postdocs of the group managed to found the permanent positions in France, and it's absolutely great. And it's I think it's my my highest achievement, because it's given the situation in France, I'm really proud of that. This are the collaborators that who participated to this work. I also would like to thank them. And I will just end up by saying that Raquel and I are creating a group called Plant Evolution here at CIBIO. And that's it. Thanks for your attention.